Thank you. I think I can safely skip some of the backgrounds here. And ultimately, we are interested in reconstructing gene regulatory networks, and it's a very old question and fundamental question in computational biology as well. Assuming if you know all the gene regulatory network within and between cell types, you would be able to find all these things automatically derived. That's why we're interested in these kind of questions. And also, we're particularly interested in causal gene regulatory networks, which are not co-expression networks, not transfer factor binding kind of networks, and not how you try to predict gene expression level with other genes expression levels. We're actually interested in if a perturbation is applied on one gene, what is the consequence in other genes, for example, right? So to look at this question, the best way is to, of course, having these perturbations, you can directly estimate or measure them, right? So but then how do you have so many perturbations, so you can look at the gene regulation from many genes to many other genes. So we have previously looked at this question with population scale bulk RNA sequencing data. In this case, you have a mutation in your DNA, right? Because we know the mutation only directly influence nearby genes expression levels, which are cis genes, and therefore indirectly influence trans genes, distal gene expression levels. Can use this relation to look for gene regulations here. And actually, this just involves testing cis and trans eQTLs in this case. And previously, we also tried to apply it on single cell data, or single cell CRISPR screen, or perturb-seq, as you have heard in earlier talks. We are able to use these perturb-seq guide RNA present to look for gene regulations between the cis gene and trans gene as well. However, they both harbor challenges or limitations. For example, here, with bulk data, you only get bulk tissues. They have limited statistical power and not very cell type specific. And with perturb-seq data, they are normally they are on artificial cell lines, and also they are just from individual donors. So it's question how much of the conclusion can be transferred to individual primary cell types and also to the population level. So the question we are asking is, can we use population scale single cell RNA-seq data to combine these two approaches to get to hopefully to get the best of both, and therefore first perform single cell eQTL mapping, and then reconstruct causal gene regulatory networks, because each mutation can be regarded as a separate perturbation, right? So there are indeed many population scale single cell RNA-seq data sets available, tens already. But then the challenge is here. You can see, it's very well known that there's a computational scalability issue. Essentially, it takes forever to map all the cis and trans eQTLs. So to think about this question, we can find a very simple question. If you just compute the Pearson correlation between all the variants and all the gene expression levels, for example, you have 10K genes and 5 million SNPs. And you have with single cell, you have, for example, 200K cells, and that is, a huge number of multiplication. And the reason behind it is, if you think about the raw data you have, actually the genotype data is indexed by donors, but the expression data is indexed by cells. And therefore, if you want to compute the Pearson correlation, you need to first expand the genotype matrix into individual cells and then multiply them together. However, actually, this is equivalent to doing a much simpler operation by first reducing the expression matrix to donors and then multiply them together. Essentially, just sum all the entries per donor in the expression matrix and multiply them together, you will get exactly the same result. And in this case, as you can see here, we have an exact algorithm that doesn't rely on any approximation, but then it essentially reduces 200K cells to 100 donors and reducing the multiplications a huge amount. Of course, we also have other optimizations here. Of course, doing Pearson correlation is a simplification. In reality, for single cell, eQTL mapping, we need to do a linear mixed model. So this comes from this, unremarkable term, which is in linear regression or in Pearson correlation, that is normally regarded as having independent error terms between individual samples. However, we perform single cell eQTL mapping because you have cells actually coming from the same donor, so the color is by donor. Actually, they will share exactly the same genotype. In that case, because the gene expression is a polygenic trait, in the sense that it is influenced by more than one genetic variant at the same time. And therefore, when you test just one variant, genotype here, the other variant will also influence the gene expression level. And therefore, cells coming from the same donor would have correlated here the error term. And therefore, this should be accounted with linear mixed model, which is very well established in GWAS. I will not go through the technical details, but this normally comes with several steps to handle, to deal with the computation. And you estimate that, genetic relationship matrix and so on. And without our acceleration, which is applies a matrix operation level, besides multiplication, but also an addition and eigen decomposition, for example, you will get a huge computational burden. But with our acceleration, and you can see here, the computational complexity essentially reduces and the number of donors, and you don't find the number of cells at all in this case. And this would completely reduce single cell eQTL mapping from single cell speed to actually pseudo bulk speed, and then it's order of magnitude acceleration. And this is the data sets we're using, and this is a very simple data set. And the main reason we use it is directly downloadable and with minimal processing. And we also simulate data with known ground truth, so we can evaluate computational speed as well as the statistical performance. So in this case, we compare with cell-rec-map. So cell-rec-map is an establishing method specifically for single cell eQTL mapping, as well as cell type specific or other context specific eQTL mapping as well. We only compared with this one because first it's work in progress, and also this is the only one for frequentist test, which gives summary statistics that you can directly use for other tasks, like in our case, performing causal inference in generic networks. You can see here, if you first look at the bottom, actually, cell recognition is relatively slow, and when we apply it on realistic size, data sets, it doesn't finish. So we have to first use it on a very small data set above, on the first row, to estimate, to know how it scales with different dimensions, and then extrapolate to get the real running time. In this case, we get 8 orders of magnitude faster with Airqtl. And normally they would take thousands of years, but essentially they use thousands of cores and taking 1 year, for example. In our case, we just need 10 minutes, for example. And of course, besides single cell eQTL mapping, another very important and very interesting question is to look at context-specific eQTLs. And that is whether, for example, context can be cell type, cell state, or different stimulation conditions. In this case, we're wondering whether one genetic variant has, of course, it may have an eQTL effect on the nearby genes in one condition, but then whether that effect would be different under a different condition or cell type, right? So in this case, we can also benchmark its speed. Of course, our method, Airqtl, is also order of magnitude faster, although smaller than in previous cases, but still way faster than cell-rec-map. Then we can look at the statistical performance, and it normally comes down to 2 questions. First, type 1, type 2 error rates, and also effect size estimation. So when we look at single cell eQTL mapping, they're generally good and comparable. But however, when we look at context-specific eQTL mapping, and that comes a challenge. And namely, if you look at the uncollaborated or the raw p-value, you find a strong enrichment, very small p-values from the now p-value. Non-eQTLs are not specific eQTLs coming from the simulated data. And of course, why the challenge is really happening, then we look at the effect size estimation, and we believe that is actually where the root cause is. So if actually effect size estimation is traditionally challenging when you are doing single cell differential expression, specifically because it's very hard to estimate the variance in the single cell expression level. And if you look at if you look at this plot, this is just you can plot the true local change or true effect size versus the estimated one, and the dashed line is the linear estimate. In this case, we find normally it happens to be we tend to underestimate the effect size and variance, and however that also depends on the expression level. For lowly expressed genes, you tend to underestimate it even more. And therefore, although in this case Airqtl does it better than than cell-rec-map, ideally want to do 1 throughout. But however, Airqtl does it better, but still not perfectly. In this case, imagine you can one gene that's just have different expression levels between 2 cell types, but the eQTL effect size is actually the same. However, just because you have different expression levels, that will lead to different amount of underestimation bias in the effect size, and then to give rise to a false positive in the context-specific eQTL mapping. And we believe this is what we see. And unfortunately, that error will propagate to downstream analysis, such as network inference. And to account for this, it's very well established to use p-value collaboration with better distribution, which here just do similar things, but however the bias is dependent on expression. So we have to parameterize the better distribution with its expression level like this. And then after collaboration, we can see the context-specific eQTLs give a relatively better distribution, and then now condition satisfied. And of course, all these two lines are drawn from Airqtls, because for cell record it never completes its context-specific eQTL to have sufficient data points allowing for the evaluation. And therefore, our speed allows it to, reveal hidden challenges and account for them. In the end, I just touched briefly on the causal inference because we can use the SNPs to find cis and trans genes and therefore to illustrate their regulations, and we can then use them to map the causal gene recognition networks. First, we remove the pleiotropic SNPs, and then we have some further constraint to improve its robustness. We need it to be confirmed by at least two SNPs. And here you can see this is the network we generate. You have a lot of genes, and it's hard to interpret it. But however, here we have we show it specifically for the flu virus activated CD4 positive T cells. These edges are shown in red, and the shared one with non-stimulated T cells are shown in black. And you see here, most of the edges are in red, indicating actually here is a very cell-type specific network, which are not observed in other conditions. And we also, to understand this better, we just look for the master regulators with the most number of targets and perform geo-enrichment to see, whether the GRN represents their actual function. I found this is indeed the case. All the top one geo-enrichment is the defense, response to the virus from, different master regulators for those that's activating or those repressing other genes. So in the end, it's still a work in progress. We are, these natural mutations are very powerful to look for, the gene regulation and to understand, to reconstruct gene replication networks. And, it can be something that hopefully, supersedes bulk population scales in RNA-seq data, and somewhat, hopefully, some tasks for, supersedes the perturb-seq data as well. And we have a novel algorithm accelerated, bringing many capacities like benchmarking, improvement methods, and as well as reconstruction European networks. In the end, because this is a very fundamental algorithm improvement for matrix operations, therefore we expect it to be extensible to other QTL mapping at single cell level as well. And initially, I wanted to have a knowledge slide, but however, I just started a new lab, so this is the whole lab is already standing here presenting it to you, so I'll just replace it with the advertisement. Thank you.
Maybe one question. Okay, maybe I'll ask the question. Yeah, very interesting talk. So to establish directionality in your causal GRN, so you're using the cis gene variation and then using that to establish the edge, right? How clean is that mapping of cis gene association? Is that like noisy? I mean, and don't you have to be like in a particular neighborhood?
Sorry?
Is that a noisy estimate? Like, how precise is that? Because, you can make really precise perturbations with things like perturb-seq.
That is a great question. It comes down to several things. First, the amount of data, whether you have sufficient number of individuals and sufficient genetic variations and sufficient cells for that. In this case, of course, there's a single cell data. It consists of different cell types. So here, we just use those with most cell types, cells. So in this case, it's about 70K each from 100 individual. And that allows us to see, I didn't personally count them, but it's allows us to have some very good single cell eQTLs in the end, but for cells with much fewer, cell type with much fewer cells, it's actually indeed pretty challenging. And ultimately, about noisiness, it also comes down to the type 1 error rate, or 1-to error rates and effect size estimation. For just single cell eQTL mapping is pretty great, but for the context specific ones, we still need a lot of progress need to be made regarding statistical methods, I would say. So, but however, that's why eQTL is here. It provides the acceleration, allows us to better understand and improve it, I would say.
Can I ask one other question?
Yes.
So have you looked at the direction, when you looked at the directionality, have you seen whether the regulators, the guys that have the outgoing edges, are they transcription factors? Like, do they have a regulatory transcription factors? Do they have a regulatory role?
Yeah, that's a great question as well. We have not looked into it. It's still a work in progress. However, this is definitely something we wanted to look into. But besides, I believe definitely some transfer factor. Probably stats-wise transfer factor, for example, I haven't looked into it personally. But however, the advantage of doing this causal inference is we don't need to restrict ourselves to transfer factors. There can be other mechanisms that regulate other genes as well.